And so I'm here not as Dr. Jeremy Brown, who's standing right over there, but I'm here to just sort of say a few words about this series. This is something that's actually sort of been a long time coming for the department. Computational biology is a place that one could argue is a department we have been sort of behind the curve on. It represents an area that in the next 0 to 20 years, you're going to see huge amounts of change. We've figured out lots of ways to collect mountains and mountains of data really quickly. And if you look at all the biologists who are sort of my age or older, which is a lot of our faculty, we were trained to spend all our time thinking about how to collect those like 5 really important pieces of data, and analyzing it turned out to be really easy. And really recently there's been this big shift. We've figured out ways to collect mountains of data really easily, and we're now spending a lot of time sort of struggling with how we really analyze that and what are the right questions to ask and how do we ask those. And that's where sort of computer science and biology suddenly come together. Are there ways that we can come together to really deal with what's going to be both a huge opportunity and a huge problem? And I'd like to thank both Dr. Brown and Dr. Belinsky for putting this seminar series together, because it really is a sort of a first step for us in trying to get people to think about what are the opportunities, what are the interesting questions, how do you handle this. I looked at the sign-up lists. For those of you in computer science and math and physics, you got to go back and talk to your colleagues and tell them that they need to be here, because you guys are totally outgunned by biologists at the moment. And so that's good from a biology perspective, but it's maybe not so good from a computational perspective. So for the next one of these, I'm expecting to see a lot more computer science, computer engineering, physics, and math people, because they've got some part of this conversation. So with that as sort of an introduction, let me turn it over to Dr. Brown. I'm going to leave, but recognize that's not because I'm not going to listen to this, but we're broadcasting this to a couple of remote sites, and we're trying out one new piece of software, and so I'm going to go to my office and listen to this remotely so I can try out this piece of software.
All right, can you guys hear me okay? Is that too loud? Maybe a little bit. I kind of feel like an astronaut here. I'm double mic'd. So yeah, as Bill mentioned, this is a seminar series that we're starting for the first time. We're really excited about it, and obviously the focus of it is on the intersection of computational techniques and biology. And so we're going to have 4 different seminars throughout the semester, once a month, usually on the first Monday of every month. This one got delayed a week because of Labor Day, but you can schedule the next 3 right now, the first Monday. And because we're going to talk about computers and biology, I'm going to encourage you to advertise on our behalf via your computers and your mobile devices, either as the talk is going on or later when you go home and you remember that really cool thing I said. You're like, oh, that was awesome. I got to tell all my friends about it. So if you're on Twitter, if you want to tweet anything about this talk as we go along, you can use the hashtag LSUCompBio. And if you want to reference me, that is my Twitter handle. So I'm going to talk about some stuff that I do in my lab and how it is biological in nature but involves a significant computational research component. So first off, why computational biology? So Bill laid this out perfectly. I think we've developed wonderfully efficient ways for collecting massive amounts of data in the last 5 to 10 years in biology. And so the buzzword, both in biology and in many other fields, has become big data and the data deluge. So we now have mountains of data of various forms. The kind of data that I think about most often is sequence data. So I'm really interested in using genomic and genetic information to trying to understand evolutionary history. So certainly the way we collect sequence data has radically changed in the last 5 years, and we really just don't know how to handle it. So if we look, just at the human genome, we're talking about 6 billion bases in a given human genome, corresponding to somewhere between 20 and 30,000 genes. We're rapidly decreasing the cost of sequencing a human genome, or any genome for that matter. And if we store it efficiently, so in other words, if we only store one copy of every base in a genome, in the human genome rather, on disk it's about 500 gigabytes. But it turns out that the way we usually generate these genomic sequences is to resequence much of the genome many, many times. So in fact, what we end up with is several terabytes of data for every genome we try and sequence. And when you start trying to do anything comparative where you're sequencing more than one genome, suddenly you have many terabytes of data. And you're trying to learn from that mountain of information. And that turns out to be a non-trivial thing to do. And you can't do it by hand, of course, so we need to involve computers. This is true not just for gene sequence data, but also for many other types. Through remote sensing, various proteomic techniques, in all fields of biology, there is and will be a great need for computational biologists, bioinformaticians, and whatever other term you want to apply to the intersection of biology and computation. And that's true whether or not you're interested in a career in industry, in academics, or in healthcare. So why these seminars? So we've said why we might care about computation and biology, but why these seminars in particular? Well, I think the way that most undergraduate training goes in terms of the classes that everyone is required to take and the kinds of experiences that you have, generally biology and computer scientists or engineers or physicists sort of have independent tracks. And at least in biology, I know that there's not a lot of formal training in computational approaches. And so the idea behind these seminars is not really to teach you everything there is to know about computation or biology, because that's impossible, obviously, in four talks a semester. But really it's to spark your interest. We want to get you interested if you're a biologist in trying to gain computational skills, and if you're in another field, computer science, physics, etc., to try to be motivated to use the tools that you have to address biological problems, because there are many and we need help. So if we have biologists over here and computer scientists over here right now, we sort of have a missed connection. And the idea is that we're going to try and marry these two fields a little bit better. So we hope that at the end of this talk and at the rest of the talks this semester, that you're interested in trying to learn more about whatever discipline you're not currently majoring in. I need to give a big thank you to those sponsoring these seminars, so in particular the Department of Biological Sciences and the Center for Computation and Technology, or CCT. They're the ones who provided the free food, so I'm sure you'll want to thank them. Also the LSU College of Science and the LBRN, or the Louisiana Biomedical Research Network. Kathy Traxler at CCT and John Cubido from LBRN have been hugely helpful in getting this all set up and going, as have a number of other folks in the College of Science, Computer Science, CCT, and the Office of Research and Economic Development. So if we could just take a moment right now and just thank them for their help, I'd really appreciate it. All right. So just to give you an outline for the rest of the semester, I'm obviously talking today. In October, the other host of this seminar series, Dr. Michael Brylinski, will be giving a talk. And then in November, Maheshi Dasanyaki, and in December, Cameron Thrash will be speaking. And they're also assistant professors in the Department of Biological Sciences who have a significant computational component to their work. And I think they're all here today. Would you guys mind raising your hands? So yeah, if you see them around, you can talk to them afterwards, or you'll see them later in the semester when they give their talks. And if you want to keep track of what we're up to, you can check us out here at this LBRN webpage. So just a little bit about me before I get started. As I said, I'm an assistant professor in the Department of Biological Sciences. I'm a computational evolutionary biologist. And in particular, I'm interested in phylogenetics and molecular evolution. So I'm interested in taking genetic and genomic data and trying to use it to learn something about evolutionary history, and at the same time trying to use the context of evolutionary history to understand something about how genes and genomes have evolved. I'm also a part-time herpetologist, so I do some field work on hybrid zones of frogs. And I mention this partly as motivation, because I didn't think I had any interest in computational biology when I was majoring in biology as an undergrad. But as I started to think about the kinds of projects I wanted to do to study reptiles and amphibians, I realized that a lot of the questions I wanted to address, the methods for addressing them in a robust way just weren't there. And so I got motivated to try and do more work on the methods so that when I actually went to gather sequence data and to learn something about these frogs and other things, that I would be able to answer the questions that I wanted to ask. And it turned into a career-long sidetrack in computational biology. But hopefully this will provide some motivation to those of you who have biological interests but not necessarily much background in computation, that you too can get involved in computationally oriented research. So just to give you a little bit of a sense for where we're going to go, I'm going to talk about the biology of the kind pertaining to the type of research I do. So we'll talk about what phylogenies are and why I think they're interesting. I'll then give a couple of case studies that I think sort of give polar opposite. They help illustrate the diverse kinds of questions that you can address using phylogenetics. So we'll talk about a bizarre ant species that I helped place find its place in the tree of life, and also the use of sequence data to try and understand the transmission of HIV in a criminal context. I'd also mentioned applying this, talking about turtles tonight, and my lab does work on turtles, but I realized in putting the slides together that if we wanted to get out in a reasonable amount of time, I wasn't going to be able to talk about all three. So if you're interested in turtles, feel free to come talk to me, but we'll skip it for tonight. After laying out the biological motivation, we're going to talk about why we need big computers to try and address these kinds of questions. So the title of the talk began Models, Genomes, and Trees. And I think it's always good to clarify the types of terms that we use in the talk. So just to make sure we're all on the same page, models can, of course, be applied to a variety of different things, from fashion models to model organisms to statistical models. In fact, tonight, perhaps some of you might be disappointed, but we will be talking, or I'm referring specifically to models of the statistical or mathematical sort. Genomes, it might seem like a slightly more straightforward word, but there's at least one possible confusion. So we won't be talking about gnomes named G. We will, in fact, be talking about genetic data. And trees, of course, when most people hear the word trees, they think of biological trees. And these are, in a sense, biological trees, but not the directly living kind. So we're going to be talking about trees in terms of phylogenies that depict the evolutionary relationships among different species or genes. So what is a phylogeny? Well, it comes from the roots phyle and genesis. So phyle pertaining to tribe, or phylon pertaining to race, and genesis pertaining to origin. So the origins of new tribes or races. Really, the way we use it now is to think more about the origins of new species. And if we had to give it a definition, it would be something like the pattern of relatedness among evolutionary units, such as individuals, genes, populations, or species. So we often think about it in terms of species, but it can often be applied as well to biological units above and below the species boundary. And a phylogenetic tree in particular is a graphical way to represent this evolutionary history in a tree-like diagram. So to give you a sense for what these phylogenies look like and how they arise, how we end up with these tree-like patterns, actually, just to give you some historical context, so phylogenies were fundamental all the way back to Darwin's description of evolution by natural selection. So this is one of the earliest sketches he did in one of his journals, and I love to point this out in all of my talks. Those of you in my evolution class who are here just saw this earlier today. But it turns out that the only figure that Darwin chose to put in the origin of species was actually one of his first depictions of a phylogenetic tree. So it was important to him all the way back then, and it's certainly important to us today. Now we can think about how a branching diagram of evolutionary relationships among species arises by beginning really at the family level. So this is an example I took from a great website called the Tree of Life website, where you can explore the full tree of life. And in this example, we're going to focus on a tiger beetle family, and we're going to focus on one individual named Mimi. And Mimi has siblings, so each row here represents one generation. And the green lines here represent lines of descent. So the two circles in the previous generation are Mimi's parents, and of course Mimi's parents have passed on their genes both to Mimi and to Mimi's siblings. Now we can think about that family couched in terms of a larger population. So we can think about Mimi and her siblings here, but of course they're not the only tiger beetles that exist at some point in time. There's actually many, many other individuals in this population, and there's complicated patterns of descent among all the individuals in that population. And we can think about this population extending back more than one generation previous. So now we're looking at least 2 generations back in the past. But we see that there's still genetic connections between everyone there. And if we start stepping back even further, we start to think about maybe time scales on the order of decades, the population here, and the green lines start to blur together. We see that there's sometimes stronger connections between certain parts of the population. There's sometimes breaks in gene flow. But ultimately, we still have one connected unit here. And really, that's how we think of species. We think of species as one connected unit of genetic descent. So they all share a common gene pool. Now, if we keep going back further, we can think not only about Mimi and her relatives, her population, and the species in which they're found, but if we go back far enough, millions of years now, we come back to the point in time where the genes found in Mimi and her relatives, her conspecifics, as we say, the other individuals in her species, where they actually share an ancestor with another closely related species. So all the genes flowing here represent an ancestral species that for some reason, maybe some geographic barrier arose and it divided 2 different populations of tiger beetles. This population went on sharing their genes among each other, as did this population, but at this point they're on separate evolutionary trajectories. And over time, they're going to start to develop various differences in their morphology, in the way they look, and in the way they behave. So this is what we refer to as a speciation event. And there's a lot of debate about exactly when in this process you call something a new species, but we can all agree that eventually, over time, things become divergent enough that they can no longer reproduce with one another, and we have 2 distinct species. So this represents sort of the fundamental unit of a phylogenetic tree. It's a branching point at which 2 species diverge from one another. And if we keep going further, we can think about these 2 species couched within a larger phylogenetic tree of other tiger beetles. We can see some of these lineages go forward through time for a while, but eventually die out. The populations no longer make it to the present. But we can think about this species couched in this broader family or genus of beetles over the course of, say, 5 million years. And eventually, if we go back far enough, we're talking about the entire tree of life. We're talking about how that genus of tiger beetles is then related to every other living thing on Earth. And this is really what phylogeneticists are fundamentally interested in. How do we understand and reconstruct the tree of life? So just a little primer on how we read phylogenetic trees. So if we have a tree structure like this, where we have 4 species called A, B, C, and D, time is read from left to right in this case. We can turn the tree so that it's not always left to right, but it'll always run from this point called the root towards the tips of the tree. And the root represents the oldest part of the tree. So this is what we call the most recent common ancestor of all the species in this tree. So this is the last time that A, B, C, and D all shared a genetic ancestry. Now we can think about this tree, we can think about the genetic changes that have occurred along each of the branches of this tree. So C, of course, diverges earliest, and maybe it accumulates 6 genetic changes. But these changes are going to be specific to C. They're not going to be found in any other species other than C. But genetic changes that happen along this branch, so this first speciation event gives rise to C, but it also gives rise to this other lineage, which is the ancestor of A, B, and D. And there's going to be some changes that take place along that branch in the tree. And then there'll be another speciation event that leads to D and to the ancestor of A and B, and then eventually this one will split to lead to A and B. But it turns out all the changes that occur along these branches right here, just below the species names, we call these terminal branches. Those are changes that are specific to each of those species. They don't tell us really much of anything about how that species is related to other species in our tree. But those changes that occur in the ancestors of species are then inherited by their descendants. So the changes that occur here we should find in both A and B. And then if we sequence the genomes of A and B, we can recognize these shared similarities, and we can use it as information to try and reconstruct this tree. And of course, the same will be true for the changes that arose on this branch and were then inherited by A, B, and D altogether. So ultimately, what we get out in the end when we actually go out and sample genetic information from various species is something that looks like this. And this is what we call a multiple sequence alignment. And in this case, it's for a single gene. Each row in this alignment, or this matrix, represents some sample we've taken. So these could be various individuals from the same species. They could be one individual from a number of species in which we're interested. But in any case, each of these is the sequence of a gene taken from one individual. And each column of this matrix, or this alignment, is a site. So it's an individual position in a gene, or in somewhere in the genome. Each color in this matrix represents one of the 4 bases. You'll notice there are 4 colors here, A, C, G, and T. So those species or those sequences that share colors have the same base at that position in the genome, and those that have another color have a different base. So fundamentally, this is the type of information that we use to try and reconstruct phylogenies. Now, this is the end of sort of the intro to phylogenetics, but I just want to point out that if you're interested in learning more about this, there's a huge, absolutely huge number of resources. There's a number of public efforts to try and make information about the Tree of Life available online. So if you search Tree of Life, Open Tree of Life Project, things like that, you can actually browse through the entire tree and see how everything is related to everything else. If you're interested in learning more about phylogenetics and thinking about trees, this is a great book and a wonderful way to get started. So I thought now we'd turn to a couple of projects that I've been involved with in graduate school and postdoc, and then here at LSU. And I think they illustrate nicely the different kinds of biological questions that we can address from a phylogenetic perspective. And the first one really is in many ways serendipitous. So this is a project that started when a friend of mine in grad school, Christian Rabeling, came to me. And Christian is a myrmecologist, so he studies ants. And he had a particular group of ants that he was really interested in. And so he's in the Amazon. He's peering at soil all day. It takes an incredible amount of patience to be a myrmecologist, I think. He's looking for these particular kinds of fungus gardening ants that he's studying. And in the meantime, he sees this small, tiny little ant that he doesn't recognize crawl across his sort of field of vision. He had cleared out the leaf litter, and he sees this little thing making its way. He didn't know what it was. Being a good taxonomist, he collected it to try and learn about it later so he could improve his ability to identify ants. So he pops it into a little Eppendorf tube, the same kind that any of you who work in a research lab know very well. And he puts it in his pocket, and then he nearly forgets about it. And he almost sends it through the washing machine. And then at the last minute, he remembers it, pulls it out, puts it under a dissecting scope, and he sees this guy. This might not mean much to you, but to a myrmecologist, this is bizarre. There are no ants that have this kind of body plan exactly. So this thing is eyeless. It's got these large pinchers on the front, and it's got this incredibly slender body, and it's incredibly small. I still have no idea how he saw this. If you clipped the nail on your pinky finger and it fell on the ground, that's basically about the size of this ant. So you can imagine sifting through something that looks like this to try and find an ant that big is pretty challenging. So Christian got really excited about this. And he, in particular, got excited about it because his old advisor, his previous advisor who had studied ants in the Amazon for a long time, had found 3 ants that looked something like this, and he had told Christian about them. And in the process of trying to collect DNA from them, the ants were destroyed, and so he was never able to actually describe them as a new species. And so Christian was very excited to find this ant again because he thought it might be the same thing that his previous advisor had found. And so he really wanted to make sure that this was something new and not just something that he was overlooking. It so happened at the time that he was doing some work in the lab of this guy right here. Does anybody recognize him? Maybe? Does anybody recognize that name? No? His name's E.O. Wilson, or Edward Wilson. He's one of the most famous biologists in the world. He's at Harvard. He's the world's authority on ants. He's sort of founded several fields of evolutionary theory relating to social behavior in ants. He's also a native of Mobile, Alabama. And he was I forget exactly how old he was, I think around 12. he was the first person in this country to discover imported fire ants. Turns out they came in through the port of Mobile, and he, as a kid collecting ants in his backyard, was the first one to find them. And he's clearly gone on to do great things since then. But it turns out Christian was working in his lab at the time, and Christian took this ant to this guy named Stefan, who's Ed Wilson's sort of right-hand man. He's one of the best ant taxonomists in the world. And Stefan didn't know what it was. So Ed is in the middle of taping a documentary for the Discovery Channel or something like that. Stefan calls him over. He makes the camera crew wait. And he walks over and he takes a look at this thing, and he's totally amazed as well. And he says, well, if Stefan doesn't know what this ant is, it has to be from Mars because he knows all the ants on Earth. And so it turns out that actually became kind of a formative moment. And so that actually gave rise to both the family, subfamily, and genus name for this ant, which is Martialis, or from Mars. And then the species name is Eureka, meaning Eureka, we found it again. And it turns out that this ant was not only cool because of the crazy story involved in its discovery. This was really convenient timing because right at the same time Christian found this ant and was able to get some genetic sequences from it, the first comprehensive ant tree of life was published. And so this is an outline of that ant tree of life. Right here we have what we call outgroups. These are closely related species to ants, but they're not ants, so these are actually wasps in this case. Turns out ants are derived wasps. And these are the various families of ants. And don't worry if these don't mean anything to you right now. But this encompasses all living ants on Earth. So we took the sequences that Christian was able to collect from, he clipped off 3 legs off this incredibly tiny ant and got some DNA out of it. I don't even know how, but he did. We took those sequences, we then compared them to all of the living ants. And it turns out that what seems to be the phylogenetic placement of this brand new ant is right at the base of the ant tree of life. So what this means is this ant diverged from all other living ants on Earth before any of them diverged from each other. This is essentially the oldest lineage of living ants on the planet. And guess how many there are? 1. Presumably there's more out there, but we only have 1 in any collection in the world. There's a single ant that's this big in a collection, yet it represents millions of years of independent evolutionary history. So it's pretty remarkable for that reason. And so it actually got a lot of attention when it was described, partly because we called it, or Christian called it the ant from Mars. So it was all over the news. It was on Fox News, MSNBC, The Guardian, Wired. Dave Barry, the comedian, even blogged about it. There were also a lot of angry comments on various news sites from people who actually thought ants had been discovered on Mars. So there were a lot of disappointed people. But so this ant is cool not only for its phylogenetic placement, but it's also cool because it really gives us a different view of the early evolutionary history of ants. So as I said, this thing is eyeless. It turns out many of these other early diverging lineages of ants are also eyeless or have reduced eyes. So here, red branches represent groups that don't see very well or don't see at all. And it turns out that many of the earliest branching groups of ants seem not to be able to see very well. And this is very surprising because the only fossils we have leading to ants are fossils that look like wasps. Again, ants are derived wasps. And these things look like wasps. They have huge eyes. They're visual predators. Their visual sense is extremely important to their lifestyle. So this suggests that there was a fundamental shift in the way that ants lived, especially early on in their evolutionary history. Now this occurred about, I forget the exact time scale, but I think it's on the order of 250 million years is roughly the base of the ant tree. It turns out there was another important thing going on in the tree of life at that time. Are there any botanists in the room? By any chance? None? So it turns out this is right about when flowering plants were diversifying. And so they were fundamentally changing the landscape. Suddenly there was leaf litter where there had been no leaf litter before. And so there was an entire niche that opened up, and it seems like these eyeless or nearly eyeless ants exploited that niche early on in their evolutionary history. So it gives us a fundamentally different view by placing this ant at the base of the ant tree. It gives us a different view of the lifestyles of early ants and how they diversified. Now, the scale of phylogeny that we've been talking about with the ants is, as we just talked about, on the order of millions of years. And that's sort of usually how people think about the tree of life. We think about genetic changes that have accrued over very long spans of time. But it turns out that doesn't have to be the case. If you have a very rapidly evolving organism, one with very short generation times, it turns out that you can get branching events and genetic changes that accumulate in lineages on the order of days, weeks, or even months. And this is especially true for viruses, and in particular, a class of viruses called retroviruses. Retroviruses normally store their genetic information as RNA, and it turns out that it's a very error-prone. They have a very error-prone mechanism for copying their genome, and so they accumulate mutations very quickly, and they have a very rapid generation time. So we can actually observe significant amounts of evolutionary change very quickly. And this becomes extremely useful in certain contexts. It's obviously important if you're interested in epidemiology and understanding the transmission of diseases. It turns out it can also be important in criminal investigations and in forensics. So there are a few cases where individuals have been accused of infecting others with HIV, either negligently or maliciously. But it turns out because HIV in particular evolves so fast, you can't actually do what we normally do with DNA fingerprinting. So when you think about DNA fingerprinting, you think about some individual who's committed a crime and they've left some biological sample at a crime scene. So what we're doing is we're comparing the genetic information in that sample they've left behind to their own genome. But because that's the genome in whatever material they left behind is the same as they carry, we can just look for identity. So we can look for exactly the same gene sequences. With HIV, you can't do that. It evolves so fast that the infections in two different individuals look completely different. And in fact, the viruses circulating within a single individual often look very different from one another. So we can think about how we might use phylogenetic information to try and understand the transmission of HIV and solve crimes. If we think about some point in time before we have a transmission event. So this is a phylogeny relating all the viruses, all the HIV lineages from a single individual. So at some point in time, that individual was infected by a single viral lineage, and that lineage diversified through time, accumulated changes, and now we actually have a diversity of viruses circulating. But they have a particular pattern of relationships among them. And so that's what this tree represents. Now, if we think about some transmission event, it turns out HIV has this property where it undergoes what's called a severe genetic bottleneck upon transmission. So rather than all of these lineages being passed on to a recipient, usually only a single lineage goes on to establish a new infection in another individual. So we're not going to get a random sample of these. Instead, we're going to get a new infection resulting just from one. And it turns out we can exploit this property to try and understand how the virus is passed between people. So in this example, if we think about this lineage being passed to a recipient at some point in time, and then later in time, after the transmission event, there's been additional evolution of the virus in the new individual, in this recipient individual. There's also been additional evolution in the host, and there's been some loss of viral lineage in the host due to perhaps the drugs that they're taking or their own immune system. The infection is still there, but particular lineages have died out. And at this point in time, we have a very specific pattern of relationships between those viruses in the recipient and those viruses in the host. So what we have is, the technical term for it is a paraphyletic relationship. We don't need to worry too much about the terms right now, but just note that the diversity of the viruses in the recipient is nested within the diversity of viruses found in the host. Another way to think about this is that some lineages found in the host, say this one, are more closely related to the lineages in the recipient than they are to other host lineages. So because of this bottlenecking, the recipient viruses are just a subset of the viruses that we find in a source individual. Now, over time, we're going to get enough loss of lineages that we're eventually going to lose that nesting relationship. And now what we have just are two groups of viruses that are each most closely related to each other. So now we have a group coming from the recipient and a group coming from the source, but they all each have their own independent common ancestor. At this point in time, we can no longer tell which direction the transmission event occurred. So we could say maybe that these two infections are closely related, the viruses are closely related, but we couldn't provide evidence that the viruses from this individual established this infection or vice versa. So if we sort of do this thought experiment, considering a number of different ways that the virus could be transmitted, we end up with three different sets of predictions. So if there's been a recent transmission event, we should see a pattern like this that we just talked about, where we have some viruses from a source, some viruses found in a recipient, but those recipient viruses are nested in the source. And we're also sampling outgroups. Any good scientific experiment should have controls as part of it. And so these are our controls. These are other HIV-positive individuals, either in the local geographic area or the most closely genetically related viruses that we could find in public databases. But if these two infections are closely related, they should exclude all of these outgroups. Now we can think about either after enough time has passed, or if person 1 and person 2 have independently gotten the infection from the same source, we'll have closely related infections, but there won't be this nesting relationship that we talked about before. And if they're completely unrelated infections, and we've done a good job of sampling our controls, then we should actually find these individuals falling out in a number of different places among the controls. The controls should break up our in-group. So we actually did this. This has been done now in 3 criminal cases in the U.S. When I was a grad student, I got involved in one of these cases in Texas, where one individual was accused of lying about his HIV status and infecting several of his sexual partners. In Texas, I believe the charge is assault with a deadly weapon, where the virus is considered to be a deadly weapon. But the prosecutors and the defense in this case were interested in whether or not a link could be established between his infection and the women's infection, the putative victims in this case. So to make things as objective as possible, we were blinded to all the identities of the individuals in this case. We just knew that there were 8 individuals, and we knew that one of them was potentially the source, but we had no idea which one. And we knew which sequences came from which individual, but that was it. So we sequenced a couple different genes in the HIV genome. I'm only going to show you the results from one just for the sake of time. But we sequenced the reverse transcriptase gene. So those of you who've taken molecular biology know that this encodes for an enzyme that's involved in turning the RNA genome of HIV into DNA. But we can still use the information directly in this gene to reconstruct these relationships. And we did that, and you can see those results here. And you'll see that we find 2 patterns that are very important to distinguishing among the possibilities we outlined earlier. So number 1, we find what we call in-group monophily. We find that all of the samples from this case are closely related to one another, to the exclusion of all the out-group sequences we've sampled. So they seem to all come from a common source. And you'll notice that there's one individual whose viruses don't form one grouping in this tree. They're actually spread all throughout this in-group. And the diversity of all the other individuals is nested inside the diversity of this one individual. So this red box represents the diversity of that one individual, here labeled CCO1. And then each of these other green triangles represents individual groupings of viruses that came from one of the other individuals. But all of these groupings are monophyletic, so there's nothing else nested inside them. And they're all nested within CCO1. So we thought that this was quite strong evidence that CCO1, that number 1, all these samples did represent a common transmission chain, and that CCO1, if any individual was the source in this case, that CCO1 was the source. And it turns out, I didn't put the slide in here to show it, but it turns out at trial it was revealed, in fact, that CCO1 was the defendant in this case. And so this evidence, in combination with a lot of other evidence in the case, ended up leading to his conviction in Texas. This story, partly for the use of this type of evidence, because of its rarity in criminal cases so far, it's only been used in 3, as I mentioned, and partly because of the extraordinary story of the women in the case who found each other and essentially did the detective work to get all this evidence and bring it to the police. Because of this case was actually featured on they were interviewed on Oprah and 2020 and a few other places. So you can see, it might be sad to say, but my biggest claim to fame might be that you can see one of my phylogenies in the background on Oprah. Hopefully I'll make more of a scientific impact, and that won't be the greatest accomplishment of my career. But nonetheless, it's probably the only exposure that many in the public will get to phylogenetics. So this evidence has been used in a number of cases. And probably will be increasingly so in the future as sequencing, the costs of sequencing drop. So we've talked about the biology, and we've talked about at least 2 examples of the different kinds of insights we can get from a phylogenetic perspective. What we haven't talked about at all yet is computation. Why do we need computers to be able to answer these kinds of questions, to be able to infer phylogenetic trees from genetic data? And in fact, my lab and several other labs in our department, all the speakers this semester, I think all of us use computational resources that are provided by the High Performance Computing Group here at LSU and the Center for Computation and Technology. So this is an image I pulled from one of their websites of one of the clusters here on campus. So why do we need computing power on this scale to try just to build phylogenetic trees? It looks pretty easy, right? We can just draw them out by hand. It turns out it's a little bit more challenging than that. So the actual computing that we're doing to try and answer these questions, to try and find the tree that is best explained by the data, the thing that we actually compute is the probability of seeing this. So let's say, as we said before, these are each sequences taken from a different individual or a different species. They represent the similarities and differences in that gene across those species. What's the probability that we would see that pattern of similarities and differences across those species if this was the true set of relationships across those species? And it turns out that takes a fair bit of tedious math to do, and so we'd rather not do it by hand. We use a probability model. So what we do is we think about the various ways that the 4 bases of a DNA sequence could change into the other. So for any given base, what's the probability, like if we start with a G, what's the probability over some span of time that any particular spot in a gene or genome is going to change into a C, T, or an A? It's very similar to a simple sort of coin flipping model, where we have some probability that if we flip a coin, we're going to end up with heads or tails. It's kind of like that. We have some probability that if we start in any state, we're going to end up in any other state, but it's a little bit more complicated. We try and add some biological realism to it, but fundamentally, that's what we do. We have some model that has quite a lot of similarity to coin flipping. But the challenging thing about this is we have to compute. So that probability that I talked about earlier, it's actually called a likelihood. It has a formal name. The probability of some data given some hypothesis is called a likelihood. And so what we're going to do is we want to compute the likelihood of all of these patterns in the sequence given the tree. What we do is we assume for the sake of convenience that each of the sites in the sequence evolves independently of all the others. So what we're going to do is we're going to compute the probability of the first site, and then the second site, and then the third site, so on and so forth until we've reached the end of our sequence. In this case, we're just calling it length S. But we have to compute each of these independently. Now again, this doesn't seem too hard. In this toy example, we only have a few sites. But even for these few sites, it's actually a little bit more challenging than it might at first appear, because we can directly observe what bases exist in species that we sample. But when we think about a phylogenetic tree, we actually have to think about what bases existed in their ancestors. Did their ancestors have an A, C, G, or T? And we don't actually know that. We can't observe that. And so what we do to account for that is we actually consider every possible base that could have existed at every ancestral position in every tree. So in this case, we have what we call 3 internal nodes. So we have a root here and these 2 internal nodes. And any of the 4 bases could have existed at any of those positions. So we actually have 4 to the 3rd possible combinations of bases across just these 3 internal nodes. And this is an exceptionally small tree, only 4 species or 4 individuals. But yet we have, if I'm doing my math right, something like 64 computations that we have to do for every site. So already we're doing 64 times S computations just to figure out the probability of observing one gene sequence on one phylogenetic tree. Now again, it seems a bit tedious, but maybe it's not that challenging until we start thinking about the number of trees we have to evaluate for any given number of sequences we have. Now it's pretty easy if we have 1, 2, or 3 maybe. We're still in sort of the handful range, 1 to 3. By the time we get up to 4 species, we're already up to 15 possible trees. By the time we get up to 5 species, we're up to 105 trees. You can see you can already see the beginning of this. This is going up pretty fast. By the time we get up to 10, we're already at almost 35 million trees. I don't even know what the names for numbers of this length are. But by the time we get up to just 50 sequences that we're trying to understand the relationships among, we have 10 to the 76th possible trees that could explain the relationships among those 50 sequences. And remember, if we really want to be sure that we have the right tree, in theory, we need to compute that probability. We have to do 64 times S computations on 10 to the 76th trees. That turns out to be really hard. For those physicists in the audience, I've been told, I don't claim to know this, but I've been told that this number is roughly on the order of the number of fundamental particles that exist in the universe, just to give you some scale of how big that number is. And again, that's for 50 taxa, 50 sequences. Let's think about the tree of life. It turns out the tree of life is big, is really big, and it's a whole lot bigger than 50. So this is an example tree of life, a sort of proof of principle that a friend of mine in grad school made. He found one gene. It's called an rRNA gene for those biologists in the audience, for which he could grab sequences from all across the diversity of life, and he sampled sequences roughly in proportion to the number of species that exist in each group. So we have fungi here, animals, plants, protists, and then you'll notice these two groups, tiny groups down here, archaea and bacteria. Almost surely, as Cameron will undoubtedly tell us about later in the semester, this is a radical underestimate of the number of bacterial and archaeal species that exist. It just happens to be the number that we've named, roughly in proportion to the number we've named. So this tree in total depicts the relationships among 3,000. It turns out that estimates for overall diversity in the overall tree of life are something on the order of 9 million. So if we're already at an intractable number of trees for 50 sequences, you can imagine how difficult 9 million, evaluating all the possible trees for 9 million sequences might be. So of course, what we do is we have a number of algorithms that we use to intelligently find our way through the space of possible trees so we don't have to do this calculation for all of them independently. And there's a whole lot of research that goes on in how you do that. And in the probability models that we use to try and account for more and more realistic biological scenarios, to try and get more accurate estimates of the tree of life. For those of you who are interested, you can download this as a poster-sized PDF from here. And if you blow it up really big, you can actually see a little label up here that says, "You are here." You know, if you need something to cover the walls of your dorm room, that might be a good option. So we talked about how we do this computation for the tree of life for one gene, but of course, as we talked about, new genome sequencing technologies are around today, and they're totally changing the way that we do all kinds of biology. So now we have not only one gene, we have hundreds to thousands of genes from all across the genome. And so finding the best ways to sift through all this data, which genes give us the best estimates of any particular set of phylogenetic relationships, and if they vary in their estimates of relationships, which ones are right and which ones are wrong. There's a lot of challenging problems there. So it's a wide open field. Now for those biologists in the audience, if you're interested in these kinds of questions and you're wondering what kinds of skills you need to do this kind of work, we use a variety of different sort of programming languages and computational platforms in my lab, and I think in many labs in the department. So we write for programs that we need to run very quickly. We often write our code in languages like C or C++ that often take a lot of time to really write a good program, but when they run very quickly, hopefully. We do a lot of our statistical analyses in this wonderful open source package called R. I should point out that all of these things are free, and there are a huge amount of free training materials to learn all of these things, to learn about all of these things. So R we use for a lot of our statistical analyses. Python we use to do analyses that maybe aren't as intense. It's what's called a higher level programming language, so you don't have to spend as much time writing code, but it allows you to get functioning code much faster. It runs slower than C or C++, but it's useful in a lot of circumstances. And in a lot of the genomic analyses that people are doing now, Python has become a very important component. But perhaps the most important set of tools we need is to learn how to interact with computers, not just by pointing and clicking, but actually using commands. And to do that, we use the Unix operating system. So when any of us run analyses on high-performance computing resources here, we manage our jobs, we control our jobs, we get our data, transmit our data using Unix commands. And so if I was going to recommend a place for you to start, it would be to start down here, go around this way. And if you're going to spend your time, start with Unix. maybe learn a little Python. And if you're interested in statistics, R is a great place to go. And as I said, there's a ton of free material online for learning about all these things. And of course, if you're interested, if you spend a little bit of time learning some of these things and you're a biologist, or if you already know these things and you're interested in getting involved in biological research, I can guarantee you that you will be in great demand if you have some understanding of biology and some ability to program. There aren't enough computational biologists out there right now. I've run into this problem, and I know many of my colleagues have, that it's just very hard to find people with that right set of skills right now because there's still this divide in how many students are trained. So I would strongly encourage you to take a little time, explore these things. And if you're interested in getting involved in research, there's a number of labs in our department and a growing number of labs that do computational biology. And you'll hear more about some of the others later in the semester. So there's a lot of people to thank. I've had a wonderful lab group. Vincent Doyle is a postdoc. Brad and John have been grad students in my lab. And I've actually had several really great undergrads, some of whom have recently moved on to bigger and better things. I've had a lot of fun collaborating with people who do everything from genome sequencing. So Mike Metzger is our collaborator on the HIV project. He's a genome sequencing expert. Bob Thompson works on turtles. These guys, Emily works on frogs. Alan and Jim are scientific. They're in the Department of Scientific Computing at Florida State. So really, being at the intersection of biology and computation allows you a lot of opportunities to interact with people in a variety of disciplines. And of course, we have to thank the funding sources who've made all this work possible. So thank you for your time, and I'd be happy to take any questions. Yeah. Yeah, the 4 nucleotides of DNA. Yeah, so that's actually a huge amount of research in phylogenetics is devoted to trying to develop realistic. Oh, sorry, yeah. So the question was about the probability model relating the transitions among the 4 different DNA bases and whether or not the rates of those transitions are all equal. There's actually a huge amount of work in the field that's being done on those models and improving their realism. So the very earliest ones have made that assumption, but any model that's applied to modern data, we relax that assumption. So we actually infer the rates of change among the bases directly from the data, and we allow it to vary across different genes and different codon positions within genes, if you're familiar with those. So we can relax that assumption for sure. Yeah. Any other questions? Yeah. 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 So the question was about turtles and our work on turtles. So amazingly, it turns out that we don't actually have a very good idea until recently of where turtles fit in the tree of life. So turtles, of course, are an amniote, so they're related to reptiles, amphibians, mammals, birds, things like that. But where they go in that tree, that amniote tree, we don't actually know. So just recently, in the past year or so, the first genomic sequences of turtles have come out. So Bob Thompson right here was one of the was involved in one of these genome sequencing efforts for the painted turtle. So it's something you can find across most of the U.S. A group in China sequenced the genomes of the green sea turtle as well as the one of the softshell turtles. And so now we're in the process of trying to essentially mine through those genomic data and compare them to say, crocodilians, birds, mammals, all of those various groups to try and figure out where turtles go. So the classic thought based on skull morphology and things like that was that turtles were actually very early diverging. So they diverged essentially at the base of all reptiles. And they've been doing their own thing for a long, long time. the genomic data seem to be indicating that they're actually very closely related to crocodilians and birds. So it looks like they diverged very close in time to the time that crocodilians and birds diverged from each other. And in fact, that's why it's been so hard with, we have massive amounts of genetic data now, but it's still hard to figure it out because all those things happen really quickly in time. And so now we're in the process of trying to sort through and figure out where they go. But yeah, that's sort of where things stand. Yeah. Was that what you're asking? Yeah. Any other questions? I don't know if we have audio from other locations or if they have any questions, but I guess not. We're all good? Okay. All right. Well, thank you all for coming. I hope you come back in the future. And if you have any questions, come find me afterwards.